Pleasure, as always, to be able to speak at the Mitochondrial meeting. I am incredibly excited to be able to share the news that we are initiating the newest 743 mitochondrial disease trial in patients with mitochondrial disease and associated seizures. Of course, I only wish that we could all be together in one room in Italy, but hopefully next year that will be possible. In the meantime, I am very appreciative to have this opportunity to be able to present this presentation by video. As many of you have probably heard, API 743 is now PTC 743. Last year, bioelectrons assets, including all of the compounds and the bioelectron team, were acquired by PTC Therapeutics. So now PTC 743 and the mitochondrial disease programs are all part of PTC. For those of you who don't know PTC Therapeutics, it is a rare disease-focused global commercial biotechnology company. The company is headquartered in New Jersey in the United States, but has offices worldwide, including an office in Italy. The company was founded 22 years ago for the specific reason to develop drugs for patients with rare diseases. And today the company has a number of drugs on the market, including those for Duchenne muscular dystrophy and the recently approved drug Evrysdi for SMA that was discovered by PTC and then developed and approved by Roche. The acquisition of bioelectron and API 743 becoming part of the PTC family is really very good news for the mitochondrial disease and other rare disease communities because PTC brings added muscle and experience to rare disease drug development and will help accelerate and increase the success of our efforts in developing drugs for patients with mitochondrial disease. This slide summarizes the many different platforms of drug discovery and development that PTC has.
Can you put the full screen?
Left side of the screen, we see the deflazacort, which is in the market in the United States for Duchenne muscular dystrophy. We have which is being commercialized in Europe and many places in the world with Duchenne muscular dystrophy. And then we have other scientific platforms, including those with splicing technologies. We have a very strong gene therapy program, an inborn errors of metabolism program, an oncology program, and then importantly, of course, our BioE program, which is focused in mitochondrial disease. The focus of today will be to discuss PTC 743 and our latest trial in mitochondrial epilepsy. PTC 743, as many of you know, has been in development for many years for patients with mitochondrial disease. Its target is the enzyme 15-lipoxygenase, which regulates a very important pathway of inflammation, oxidative stress, glutathione depletion, and cell death, which is known to be important in a number of CNS diseases and has increasingly been shown to be associated with CNS disorders of high levels of oxidative stress. It is given orally, either by mouth or through a feeding tube. It has a number of orphan designations around the world and one fast-track designation. Over 500 patients have been treated around the world with PTC 743, and the longest treatment is over 10 years now in a boy with SIRF1 syndrome, who was the second patient ever treated with PTC 743 back in 2009. Importantly, as we've discussed many times, PTC 743 continues to be safe and well tolerated. And of course, we have gathered a lot of important data over the years in terms of clinical benefit as well as biochemical effect. We are proud to be announcing this trial in mitochondrial epilepsies, which I'll be detailing shortly. And we'll also be launching a phase 3 trial in Friedreich ataxia before the end of this year. The two trials in mitochondrial epilepsy and then Friedreich ataxia, which will be starting later this year. We have spent many years trying to fully understand the target and mechanism of action of PTC 743. We had a number of important clues early in its development over the years, including important clues from work done at the laboratories at the Bambino Gesù Hospital, as well as work done elsewhere, that we knew we were having an effect on oxidative stress and glutathione. Through many years of careful experimentation, we honed in on the specific target of 743, and that's the enzyme 15-lipoxygenase, which regulates a very important pathway of lipid-based inflammation, oxidative stress, and cell death. In the presence of oxidative stress, the enzyme 15-lipoxygenase gets upregulated and oxidizes polyunsaturated fatty acids. These oxidized fatty acids are potent inflammatory signaling molecules and are shown to be significant propagators of the inflammation and oxidative stress response that lead to further cell injury and cell death. 743 is able to donate an electron to the core of 15-lipoxygenase, essentially inactivating the enzyme, which we have demonstrated in a number of cellular studies that to decrease inflammation, decrease oxidative stress, and overall improve cell viability. This pathway of 15-lipoxygenase is becoming increasingly recognized as an important aspect of CNS disease pathology, including in Parkinson's disease, Alzheimer's disease, Friedreich's ataxia, as well as epilepsies, where mutations in the enzyme GPX4, which sits right next to 15-lipoxygenase in the same pathway, have been linked to refractory epilepsy syndrome. The development of the refractory mitochondrial epilepsy trial is a result of our efforts over many, many years in the laboratory and in the clinic. It leverages learnings we've made not only, again, in the laboratory, but also important learnings I think that we as a community have made about the challenges in developing drugs for patients with mitochondrial disease. We have spoken at this meeting and many others many times about the difficulty of finding endpoints that are validated, that are known to be important to the disease, and that are measurable. In addition, we know that when we try to study specific syndromes, we have challenges that related to disease heterogeneity that results from the complexities of mitochondrial disease. And so the ability to identify a symptom, seizures, that are known to be incredibly important in patients with mitochondrial disease and that are not well treated by the current available therapies, provided us an opportunity to have an endpoint that is relevant, that is measurable, that regulatory authorities around the world understand, it's validated, and allowed us to actually find something that's common to many patients despite differences they may have in specific genetics or in specific disease subtype. And so we developed this trial, again, with a strong scientific rationale about the pathway, the target pathway of 743, as well as observations and data recorded from clinical studies we have conducted over the course of many years. We have looked at PTC743 in the lab and looking at its ability to protect patients' cells from lipid oxidation-mediated cell death. This slide demonstrates potent protection of PTC743 provides in cells from a patient with POLG, from pontocerebellar hypoplasia type 6, Leigh syndrome SIRF1, basically all cell model systems where we're able to recapitulate the pathway activated by 15-lipoxygenase and show that 743 is a very potent rescue agent at nanomolar concentrations. We've done similar studies being able to show the protective effects of 743 in more relevant models, including primary neuron models, where again, we can induce the inflammation, oxidative stress, cell death pathway targeted by 743 and be able to demonstrate that 743 can be a potent rescue agent. But more importantly is the clinical reports we had gathered over many years of patients with mitochondrial disease and associated seizures. And this came from our expanded access studies, as well as other specific compassionate use trials. In our earliest treatments of patients going back 6, 7, 8, even 9 years, we had reports from physicians of patients with Alpers-POLG, complex 3 mutation, MELAS, and a non-specific mitochondrial disease mutation, that patients treated with 743 were having reduction in seizure frequency. Patients who came in with refractory status epilepticus were having resolution of status epilepticus without occurrence. and that they were overall improvements in seizure-related morbidity. We also had data from a study conducted at the Bambino Gesù in a case series of patients with pontocerebellar hypoplasia type 6, which is secondary to the RARS2 mutation, which is a particularly severe mitochondrial epileptic encephalopathy. These children typically begin having seizures early in life. The seizures persist and typically don't respond to medications. 5 patients were treated with PCH6, and I believe these data have been shown here previously. But what we did is, after all patients completed 36 months of treatment, we did a retrospective review of the treatment experience and found a number of key data points related to seizure effects as well as seizure-related morbidity. In particular, 2 subjects who entered the trial with refractory status epilepticus had resolution of the status epilepticus with the introduction of 743 without recurrence over several years. There was an overall reduction in seizure frequency recorded, as well as, importantly, a decrease in disease-related hospitalizations. As shown in the graph on the right, the 5 patients had on average 52 days in the hospital per year in the year prior to starting on 743, and this decreased to 7 days in the first year, 0 days in the hospital for disease-related complications in the second and third year on 743. We also conducted a natural history study where we did a literature-based natural history that was supplemented with reports from physicians who had treated these patients. We found a cohort that we matched for genetic severity, onset of seizures, as well as other symptoms, and were able to demonstrate that a decrease in mortality risk relative to the reported natural history in these patient groups. Finally, we also looked back at some of the Lee syndrome trials we had conducted. And while at the time we hadn't specifically focused on seizures with seizure diaries, we looked specifically at the item in the Newcastle score that looked at seizure frequency and looked basically from baseline to 6 months later following treatment with 743. And a number of patients who had the specific item of seizures with either mild or moderate seizure frequency, we showed that over the course of the study, this seizure frequency declined, usually from 1 grade, from moderate to mild or mild to none. Now, while taken as a whole, none of these data were rigorously collected with seizure diaries prospectively. What they did do is provide us with evidence of a potential important effect on the refractory seizures that children with mitochondrial disease had. And so the obvious next step was to design a more rigorous prospective randomized controlled trial where we could look very carefully and determine the benefit of PTC743 in patients with mitochondrial disease and refractory seizures. And that is the trial that we have now begun. This is a study in patients who have genetically confirmed mitochondrial disease and associated refractory seizures. The protocol was developed in close collaboration with the EMA as well as the FDA, and the protocol has been approved by both regulatory agencies for conduct. It is a randomized placebo-controlled trial with a target enrollment of 60 subjects. The study will be a 7-month study followed by a long-term extension during which all patients will receive 743. The format is like this. Patients will be screened and enrolled, and there'll be a 1-month observational run-in phase to ensure that patients have a minimum of 6 observable motor seizures. If the patients meet that specific criterion, they will then be randomized to receive PTC743 or placebo for 6 months. Then, following that 6-month placebo-controlled phase, all patients will receive PTC743 for 48 weeks as part of an open label extension. I am proud to announce that enrollment on this trial has begun. We opened for enrollment in October, and we've had the first subject screened and entered into the trial. I want to review the key exclusion and exclusion criteria. The age for inclusion is less than 18 years. You must have genetic confirmation of mitochondrial disease. It can be either from a mitochondrial genetic defect or a nuclear genetic defect with associated epilepsy phenotype. Patients must have seizure activity despite ongoing treatment with at least 2 anti-epileptic drugs. And of course, we will verify that patients have greater than or equal to 6 observed motor seizures during the 28-day run-in phase. There must also be a documented history of epilepsy, a stable dose regimen of anti-epileptic therapies, and a stable regimen of dietary supplements. And if patients are on a ketogenic diet, a stable diet for 90 days prior to baseline visit and for the duration of the study. I want to point out that this is another important learning that came from many discussions we've had with several of you at this meeting today, as well as other investigators, about how to manage supplements in the trial of mitochondrial disease. Given that this is going to be a global trial with patients enrolled in many different countries, we wanted to be considerate of the fact that practices of supplements and mitochondrial cocktails differ in different locations, and wanted to allow patients to stay on their current regimens during this trial. Quite simply, if they are still meeting the other criteria of having ongoing seizures despite being on a cocktail, we thought that that was the important point in terms of entering into this trial. The key exclusion criteria really relate to abnormal laboratory values, including those related to liver dysfunction or abnormal coagulation or abnormal kidney function. It's also important that patients not have been in another interventional clinical trial for 60 days prior to randomization, and of course for the duration of this clinical study. Patients, the 60 patients enrolled will be randomized 1-to-1 to receive vatiquinone or 743 or placebo, but we're also going to stratify by disease type. So there'll be a balance for patients with Alpers-Polg, Lee syndrome, and MELAS, and then the fourth strata will be for other genetically confirmed diseases. And we have a certain number we're trying to achieve in each of those different categories. And again, I'll just mention that that approach was something that the regulatory authorities had asked us to do. The primary endpoint of the study will be change in observable motor seizure frequency. So basically looking at the change in number of seizures during the 6-month treatment period relative to the 1-month run-in. And then we'll have a number of secondary endpoints looking at disease-related hospital days, status epilepticus, use of rescue medications, and quality of life. And we'll, of course, look at safety endpoints, including laboratory values and serious adverse events. We're looking to have approximately 15 sites globally, including in the EU, the United States, South America, and we'll be enrolling, as I mentioned, 60 subjects. We may require having additional sites and investigators in order to complete the study. Obviously, in the COVID environment, we are very aware of the potential impact of an additional wave or another period of high numbers of COVID cases and how that could impact the trial, particularly given the fact that patients with mitochondrial disease can be very fragile, and we have to be very careful of their health and safety and need to balance that with the need to get to study sites. So we're looking very carefully at the geographic locations of study sites, and what we have also done in this protocol is try to incorporate as best as possible the ability to do assessments remotely, so by telemedicine or video when possible, so that patients need to come to the study sites as infrequently as possible. I'll also note that this way that we're recording seizures in this study is by an electronic diary, so we will be giving an electronic device to all patients in the study. And so this is just one way that we can actually check the logs and make sure everything is being done appropriately and recorded appropriately remotely and not having to come to the study site. And finally, the very good news that we have begun enrollment. The first sites have now been opened in the United States. We are working to get the additional sites in the United States and the sites in Europe opened as quickly as possible so that we can complete enrollment in the study quite rapidly. We have set up a website for the study that can provide anyone, physicians, families, patients, anyone in the community who's interested, more information. And that website is www.the-MIT-E-MIGHTY-study.com. Again, thank you all very much. A pleasure to have this opportunity to share this update with you. We're obviously incredibly excited after many years to be able to be starting another 743 trial, being able to incorporate the learnings that we have made over the years in treating patients with 743, the learnings we've made in conversations and discussions with many of you over the years. We look forward to continuing our collaboration with you all and to continue our efforts to bring 743 to patients with mitochondrial disease. Thank you.